So first, I'd like to thank the organisers for inviting me here today. It's a great pleasure to come back to Imperial College. I think it's maybe it's the first time I've given a talk here since I gave my thesis defence all those many years ago. So it is nice to be back and talk about quantum biology. So the question I'm addressing is why biology needs quantum mechanics. And certainly when I was a biologist being taught by chemistry at the University of London and then a PhD, we didn't need quantum mechanics. Nobody taught us quantum mechanics because it wasn't thought to be necessary. But the first thing I want us to think about is, what is life? It's a kind of question that I think all biologists ponder. What is life? What makes it so different from all the other stuff that is in the world? Much of it composed of very similar materials than living stuff, but with completely different kinds of dynamics, much more, at least from a biologist's perspective, much more boring than life. So what is it that makes this stuff so different than that? And what we are proposing, myself and Jim, in the book we've written, is that life is kind of on the edge of between the classical and quantum world. Now, I'm going to be assuming that there are probably many physicists in the audience, but also many people who aren't physicists who don't know anything or very little about quantum mechanics. So I'm going to be talking and giving a kind of intro to quantum mechanics. But this is the world that I learned about when I was being taught biochemistry and through my career as a biochemist, the classical world, the world of astronomy, Newton's apple falling on Newton's head, thermodynamics. And the key thing, of course, about all of this is that beneath the order that we see around us is disorder. Now, quantum mechanics is different, and somewhere between the quantum mechanical world and the classical world is where I believe life sits. And in order to illustrate quantum mechanics to those people not so familiar with it, I'm going to look at an experiment that Richard Feynman said was all quantum mechanics are contained in this experiment, the double slit experiment, which demonstrates wave-particle duality, that particles can be waves and waves can be particles. So we're going to go through the experiment to illustrate it. So start off with light. Now it shows a light bulb, but really this should be a laser, because we want a single frequency of light to go through two slits. And when they do, because these waves are coherent waves, because they're coming from a coherent light source and they're of a single wavelength, when they pass through either the top or the bottom slit and then travel to meet the screen, at some points on the screen, the waves cancel each other out. The trough of one wave meets the peak of another wave, and the waves cancel out. At other points on the screen, the waves reinforce, where the peak of one wave meets the peak of another wave. So you get this interference pattern on the screen, which is characteristic of wave phenomena, not necessarily quantum mechanical. Any wave phenomena will demonstrate this kind of interference pattern. So interference patterns are for waves. Particles tend not to do this kind of thing. So if you pass sand through a double slit experiment, it doesn't form interference patterns, it forms piles of sand. No interference. So the simple lesson is single waves go through both slits at once to generate interference patterns between the alternative paths. And these can be seen not only in lots of different media. We can see interference pattern. If we drop some stones into a pond, you can see that kind of interference pattern. Particles go through one slit or the other and do not generate interference pattern. So that's how we distinguish between particles and waves, and now we ask the question, is an electron a particle or a wave? And this could be applied to an electron, a photon, an atom, even molecules will do exactly the same kind of behavior. So we have the double slit experiment, and first of all, let's close one of the slits, and we allow electrons to fire from this electron gun through the slit, and you will generate a scatter of particles, a pile of sand kind of pattern. This looks like a pile of sand. And that's what we think is doing, that the electron is passing through there. But now we ask the question, what happens if we open up the other slit? And now when we do, we get an interference pattern. So although with one slit open, the electron looks like it's behaving like a particle, with two slits, you get an interference pattern indicating the electron is behaving like a wave. Okay? And the first thing you might ask is, well, okay, a wave. Water has waves, and waves are caused by collective behavior of lots of water molecules. Maybe this waviness of electrons is because of the collective behavior of electrons. So to test that, you can fire electrons one at a time. So you remove the possibility of any collective behavior by firing electrons one at a time through the screen. And when you do, you see the electron leaving the gun, passing through one slit or another, presumably, and forming a dot on the screen. Looks like a particle. Leaves the gun as a particle, arrives on the screen as a particle. It must be a particle. But as you keep on firing the electrons through this screen, you start to build up more and more dots. And as you do so, you start to see an interference pattern. So here, firing particles individually through this double slit experiment demonstrates an interference pattern which suggests that that particle is somehow going through both slits at once. Because that's the only way we can generate an interference pattern, as a wave. So you kind of say, as a classically trained scientist like me, no, that can't be right. It must be going through either the top or the bottom slit. No question. Let's detect where it is. So we can put a detector on one of the slits and say, hey, it's either going through this one or that one. So we put a detector on. Here we are. Repeat the experiment now with an electron detector, say at the top slit. If it goes through there, we know it's gone through there. If it lands at the screen and the detector doesn't beep, then we know it's gone through the bottom slit. So we have a way of detecting where the electron goes. And now we don't get an interference pattern. So when we try to detect the electron, we no longer get the interference pattern with the electron detector on. And you think, oh, well, maybe the presence of the electron detector is somehow destroying the interference pattern. Well, let's turn the electron detector off. And you get the interference pattern again. So something very strange and weird is going on here, that the electron behaves like a wave when we're not looking at it, but when we look at it, it behaves like a particle. So it seems to be that it leaves the electron gun as a particle, then somehow travels as a wave through both these slits to generate an interference pattern at the screen. But that interference pattern is actually formed by individual particles landing or at least being detected at the screen. So somehow on its travels, it traveled as a wave, but when it met the screen, it became a particle. But if you have the detection here at the screen, then it remains a particle all the way through. So that's the way matter behaves at the quantum mechanical level. Particles have both wave or objects such as electrons, atoms. This experiment has been done with atoms. It's been done with molecules such as C60 fullerene, also generates these kind of interference patterns. You know, the kind of molecules that are of a biochemical kind of size. Amino acids have also been shown to generate this kind of interference patterns. So our vision as biochemists trained in the classical tradition of ball and stick molecules is an illusion. Particles aren't balls and sticks. We may be useful to think of them like that, but when we're not looking at them, they're waves. And they're waves not only in these experiments, but they're waves in our cells. That wave mechanical behavior doesn't go away when we're not looking at it, it stays there. So it's having implications in the way cells work. And that's an important feature. So measurement, not sure if I get into this later, but measurement is the key between the classical and the quantum world. If you don't measure things at the atomic level, if you like, they behave quantum mechanically. But when you measure them, they behave classically. You turn waves into particles. It's the most mysterious and controversial area of quantum mechanics, measurement, quantum measurement, what exactly it is. And physicists in the audience, I'm sure, will be very familiar with all of the controversy around what makes a measurement or not. And we won't be going into that, but it is a very interesting question from the perspective of whether life makes quantum measurements. So this is kind of summary then. Particles behave as waves when we're not looking at them. So they can be in two places at once. Measurement forces the particle to choose one route. If you measure it at the screen, then it chooses one route at that level. If you measure it at the slits, it will choose to be a particle at that level. And it can then be in one or the other slits, but not both. If you don't measure it, it goes through both, and it only becomes a particle when you measure it at the screen. So that's more or less all of quantum mechanics is contained in that fairly simple experiment which anyone can perform with appropriate equipment. So another way of looking at it is the skier. The quantum skier can go one way and the other way round the tree at the same time. So if skiers behaved like quantum mechanical entities, then that's what they could do. The question we have to think about is whether we're skiers like this or a classical skier. Certainly we are skiers like this, but what about ourselves? Probably also skiers, normal classical skiers, but somewhere between cells and individual particles, it's like this. And a key question in biology, at what level do we get the quantum mechanical stuff and the transition between quantum and the classical world? So that's what I've told you about. The double slit experiment is a feature of quantum coherence, which is essentially that particles are behaving like waves. And when we talk about quantum biology, we are talking about some aspects of quantum mechanics, such as quantum coherence of particles. That is non-classical. So one thing I want to get through early on is that we're not talking about the, if you like, the trivial features of quantum mechanics, like having energy levels in atoms. We all know those exist. They're entirely quantum mechanical. They are responsible for why I don't fall through the floor. So we're not talking about that in quantum mechanics. That's already accepted. We're talking about the, if you like, the weird aspects of quantum mechanics that we don't expect macroscopic objects to do. We don't expect macroscopic objects to be in two places at once. Yet quantum mechanical objects can. So that's coherence, quantum coherence. But there are other features of quantum mechanics, such as particles being able to tunnel through walls that they shouldn't be able to pass through energetically. That's kind of illustrated here. You can't see it, but some of that particle is actually passing through the wall here. And that is quantum tunneling, which allows particles, because of their wave-like properties, to tunnel through impenetrable barriers. And that's another peculiar feature of quantum mechanics, not allowed in the classical world, but also seems to be involved in life. And then the oddest feature of quantum mechanics, which I'll be saying something about as well, is entanglement. Particles can have spooky connections. Now, the spooky connections bit comes from Albert Einstein, who never quite came to terms with quantum mechanics, even though he was one of the people who invented quantum mechanics. Much of his of early quantum mechanics was based on his work. And yet he couldn't really come to terms with it. And one of his arguments was he was the first person to recognize that in the quantum mechanical world, if quantum mechanics is right, then if you separate two particles to arbitrary distances, a galaxy, a universe apart, they will still be connected. They are described by the same wave function in the physics terms. And if you tweak one of them, the other one will jump at that moment in time at the other end of the universe. And what Einstein said, no, that can't be right. And he called it spooky action at a distance, because it seemed to violate general relativity. But experiments have demonstrated many times that entanglement is real, and particles do have these spooky connections. No one's demonstrated across universes, but even across continents it's been demonstrated that entanglement exists. So entanglement is another peculiar feature of the quantum world that is not present in the classical world. Now, the early quantum pioneers, such as Erwin Schrödinger, became interested in life and the question of whether quantum mechanics played a particular role in life. This is some notes from a lecture which he was giving in 1933. These are chromosomes. He was trying to work out how chromosomes worked. And at that time, heredity was being worked out by many scientists. And he was interested in the question of whether heredity may have a quantum mechanical component to it. And then in 1944, he wrote this book, What is Life?, in which he did make the claim that quantum mechanics is fundamental to life, certainly fundamental to heredity, but also fundamental to life. And it's interesting to have a look at why he made this claim, what it was based on. This is a statement he made. There are many statements in the book, but this is one that's interesting. The living organism seems to be a macroscopic system, which in part of its behavior approaches purely mechanical as a contrasted to thermodynamical behavior, to which all systems tend as the temperature approaches the absolute zero and the molecular disorder is removed. Now, this is a long-winded way of saying quantum mechanical behavior, because when you cool stuff to absolute zero, remove the molecular disorder, objects behave quantum mechanically. So he claimed that genes behaved like a structured solid, some kind of crystal, but one that encodes information. He called them genes were aperiodic crystals, order from order. So to illustrate that, so his argument really was that the laws that govern the macroscopic behavior of objects, such as steam trains, are actually statistical in nature. So the gas laws, the diffusion laws, thermodynamic laws, they're statistical in that they depend on the random motion of very large numbers of molecules. And when you look at something like the pressure of a gas in a box, that pressure is generated by the random collisions of trillions of particles. And the laws you get are statistical in nature. And he called these laws order from disorder. So at a molecular level, there's disorder, just particles bumping around in boxes. But at a macroscopic level, you have the kind of laws that drive steam trains, like the gas laws. So he claimed that whereas you can get order, this is a famous chemical reaction which generates these structures in a petri dish, and they're structured at the macroscopic level, but if you look at them at molecular level, it's all disorder. Order from disorder. But what he claimed, that life was different, that living organisms have order all the way down. Now, this is actually an enzyme, a cartoon, an animation of how an enzyme works, so biochemists will be familiar with it, but essentially it is order all the way down. Enzymes are highly structured objects, and so is DNA, and so are the other biomolecules of life. So this is what he claimed, that in inanimate objects, there's chaos, disorder at the molecular level, and order at the macroscopic level. Life has this structure, order all the way down. That's what Schrödinger claimed. Now, he claimed this before, he claimed it particularly about genes before we even knew what genes were made of. At the time, there was a lot of controversy. They knew that genes were in chromosomes, but chromosomes contain protein and DNA, and many people thought that protein was a much better substrate for genes. So nobody really knew what genes were, but he claimed that these kind of aperiodic crystals in which atoms, particles, encode order. Now, the problem with that, the problem with why that is a kind of controversial claim, is that the quantum mechanical world requires coherence. All particle waves need to wave in step. So if you have a number of objects, remember in quantum mechanics, particles have wave-like properties. An individual particle will always have wave-like properties. Two electrons, say, will have wave-like properties if they both wave in step. The same for 3 electrons, 4 electrons, 10 electrons, 10 trillion electrons will have wave-like properties if they all wave in step. But this is where the border between the quantum and the classical world kicks in. Because as you have more and more particles involved, it's less and less likely that they will be waving in step. So quantum mechanics requires this delicate order, which is very fragile. All of the particles within an object have to be waving in step. It's easily lost in complex, noisy molecular environments. So it's kind of a way of illustrating it is that you can detect wave-like properties by dropping 2 pebbles in a still pond, but try demonstrating that water has wave-like properties in a torrent. It's not going to be so easy because the disorder of the water shatters all these waves. And that is what is happening between the atomic world of quantum mechanics and the macroscopic world we see around us. We're going from here to here. And the question is, where exactly? At what level? Now let's see if this works. Hopefully it will. So this is a animation of a cell membrane. Let's see if it'll, there we go. Now, that's an animation of what we think is going on in a cell membrane. Thermodynamics, all that moving around, all that motion, that looks pretty darn random. So we wouldn't really expect to see quantum mechanics in these kind of systems. There's just too much random motion. So although Schrödinger may have claimed that quantum mechanics was at the core of life, as scientists were finding more and more about what's going on inside cells and discovering processes that were driven by thermodynamics, they said, no, it can't be. Okay, so here a point 2. But the key question then is quantum mechanical phenomena are only to be found in simple molecular systems that are capable of maintaining quantum coherence for biology-relevant timescales. So in biomolecules, individual biomolecules, you will have quantum mechanical phenomena going on. And what Schrödinger and others claimed is that life is able to amplify these quantum mechanical phenomena, so you see the effects of them at a macroscopic level in living organisms. So these are the level of atoms. This shows the kind of structures that are present in life, atoms, molecules, DNA, proteins, etc. So these certainly behave quantum mechanically. These certainly behave classically. Where is the transition along this scale map? So the likelihood of maintaining coherence will drop as the number of particles involved in any phenomena goes up. So that gives you the kind of background of the issues involved. And now we're going to, and just to end, well, actually, we'll come back to it sometime, the Schrödinger story. So Schrödinger published his What is Life in 1944. It was inspirational to many scientists, including Watson and Crick. But after the DNA double helix was discovered and molecular biology was so successful, pretty much everyone forgot about quantum mechanics in biology. And biologists continued studying biology without learning any quantum mechanics. But more recently, there is interest in quantum mechanics because quantum mechanical phenomena have been identified in a number of different areas of biology, and we'll look at just a few of them today. So, first one, photosynthesis. Of course, the biologist, well, everyone knows what photosynthesis is about. The most remarkable reaction in the universe, the air, the stuff around us, plus sunlight, makes plants. It is absolutely astonishing. And it does it with some remarkable chemical reactions, splitting water, for example. So it's the most astonishing chemical reaction we know about. Air plus sunlight makes plants. And we eat those plants. So all of our biomass is derived, pretty much all of our biomass is derived from this reaction. So it's the most important reaction in this entire biosphere. And the evidence is coming down that it fundamentally involves quantum mechanics. So inside plants and all the other photosynthetic organisms, many of them microbes, are photosynthetic systems which consist of proteins and chromophores, such as chlorophyll. And this shows chlorophyll, porphyrin ring with a magnesium atom sitting in the middle of it. And this magnesium atom has loosely attached outer electron, which can absorb a photon. And that's the first step in photosynthesis, that an electron in the outer orbital of the magnesium atom absorbs a photon. So this is the first step illustrated here. Photon of light is captured by a pigment molecule in a reaction center in a plant or a microbe. First step involves the capture of a photon of light by the antennae pigment and its conversion to an oscillating exciton. So an exciton is just an electron that has leapt out of its orbital and then can fall back in, so it vibrates. And this is called an exciton. It's essentially a vibrating electron that's captured that photon energy. So the exciton has captured the photon energy, and then the exciton in photosynthesis, in the first step of photosynthesis, that captured exciton has to jump through a system of pigment molecules, and a very dense system of pigment molecules to find its way to the reaction center. Now, that's what they look like. So somewhere along here, a photon is captured by a pigment molecule, and what happens is it has to be transferred along a kind of, you can imagine it as a kind of pebbles in a pool that you have to walk over, and the exciton has to vibrate in one pigment molecule, and then it has to hop to another pigment molecule and another to make its way to the reaction center. But the problem, what is hard to understand is that this energy transfer reaction is extraordinarily efficient. It's nearly 100%, as close as you can measure to 100% under optimal conditions. So under optimal conditions, all of the energy of every photon gets transferred to the reaction center, where that energy is then used to fix carbon dioxide and make the plant. So it's 100% efficient, and it shouldn't be. The reason it shouldn't be is that that exciton is unstable. And its transfer from where it's picked up to the reaction center should take longer than its stability. So it should mostly fall down, the electron should fall down and release the photon again. So the energy should be lost. And it isn't. The energy nearly always meets the reaction center. So it has this 100% pretty much efficiency. And the problem is understanding how. And it's essentially what we could call a traveling salesman problem, in that the exciton could travel through this forest of pigment molecules in any direction it wishes to go. It doesn't know where the reaction center is. So it should wander through the forest of pigment molecules and then emit the photon, and the energy would be lost. But mostly it seems to find its way very efficiently to the reaction center. And understanding how it manages to do that is the problem. So, a big step forward in that was, that should be quantum beats in photosynthesis, was taken by Greg Engel, who was working in Graham Fleming's lab at University of California. And essentially what they did, and I won't go into the details of this very complex experiment, was using femtosecond spectroscopy, shining at a photosystem a sharp femtosecond beam of light, and then essentially looking at the reflection of that light as it was, as it kind of reflected out of the photosystem. So when it was doing that, it probes the photosystem and the energy transport in that photosystem. And what they found was that the energy came out in beats, in waves. Now, there isn't time to go into the details of this experiment, but these quantum beats, as they are called, this shows an illustration of the system that they looked at. It was a bacterial photosystem, simpler than the plant one, and it only has a few pigment molecules. Still has the same problem. The photon has to be captured. There are many different routes it can take to the reaction center. And what they found was it travels to the reaction center as a wave rather than as a particle. So it's like the quantum skier. It goes through both ways, or all possible ways, simultaneously. And this seems to be part of the reason, at least, why it finds its way to the reaction center so quickly. It tries all routes simultaneously. So exciton travels to the reaction center as a coherent quantum wave that samples all routes simultaneously to find the fastest route to the reaction center via something called a quantum walk. So essentially what's happening, and then Seth Lloyd, a theoretician based at MIT, suggested that as the system goes from the quantum mechanical dynamics to the classical dynamics, a process known as decoherence, is that the system is tuned to allow the wave to find its way to the reaction center, and then bang, it becomes a particle. So Seth Lloyd claimed that this happens, that the system is tuned to allow this maximal transport, what he called a Goldilocks effect, that it's at just the right level for the decoherence is tuned to allow this to occur maximally efficient. So it travels. So that's the first, one of the first experiments in the modern age of quantum biology. One of the first, but not the first, because actually, so photosynthesis seems to involve this coherent transport of energy in photosystems, and that's been demonstrated now in many different photosystems, in not only microbes, but plant photosystems, and it seems to be fundamental to photosynthesis. And as I said, photosynthesis is the most important reaction in the biosphere. So having quantum mechanics as fundamental to it is obviously very important. But the proteins that we saw, these are in photosystems. If we look at this, most of what you're seeing here is protein. Those proteins and the pigment molecules are made by enzymes. So the other side of life is enzymes. And what is known now is that enzymes, the engines of life, so enzymes, as any biochemist in the audience know, well, all biologists, I'm sure many other people know, all of the biomolecules in every organism, living organism, have been made by enzymes. Enzymes make everything. They're the engines of life. They make all our biomolecules. And they do so as catalysts. So they're catalysts. They speed up chemical reactions, but by factors as high as 10 to the 20. And you guys are familiar with numbers like 10 to the 20, but how I illustrate it to if you like, secular audiences, if we could speed up our walking speed by 10 to the 20, we could walk across galaxies in milliseconds. That's the kind of speed up that enzymes deliver to chemical reactions. And it's difficult to account. There are many different mechanisms involved in enzymes, as many as the biochemists in the audience will know. So we can get that to move again. But the first thing we should notice about how enzymes work, and this is an enzyme dihydrofolate reductase, and what you see is the substrate coming in and being manipulated by the enzyme to generate a product. What you ought to bear in mind is that these are single molecules full of individual particles, and what the enzyme is doing is manipulating matter at the level of atomic particles. So it's bound to be bumping into quantum mechanical phenomena. What has been discovered is that it is using quantum mechanical phenomena, that how enzymes work in their active center, grabbing substrate molecules, manipulating them and turning them into products such as the proteins in photosynthesis, such as making DNA, such as making chlorophyll, is that they utilize quantum mechanical phenomena, particularly quantum tunneling. So first that was discovered actually back in the 1970s, that in electron transport chains, such as take place in our cells in respiration, where electrons from respiratory substrates are fed into electron transport chains and transported along the chain to eventually be dumped onto oxygen as the terminal electron acceptor. The electrons, that is a transport rather like we looked at in photosynthesis, but instead of an exciton being transported, it's electrons. And what was discovered is that electrons are transported by a process of electron tunneling. And this is an entirely quantum mechanical phenomena in which electrons can, or any particle can be transported from one place to another through a barrier that would be classically impenetrable. So it could be a very high energy barrier, for example, that the electron shouldn't have the energy to jump over, and it can tunnel through because of its wave mechanical properties. And this is responsible for how electrons are transported very efficiently along electron transport chains in the respiration that keeps us alive. But more recently, it's been shown that protons are also tunneling in enzymatic reactions. So this is another enzyme AADH, aromatic, I can't remember what the other AA is, dehydrogenase. So this shows the enzyme active site. And in order to probe whether or not quantum tunneling is involved in these reactions, in this case, quantum tunneling of protons, the nucleus of a hydrogen atom, you can do a useful trick, and that is to replace hydrogen with deuterium. So quantum tunneling is essentially going through an impenetrable barrier through quantum mechanical phenomena. If hydrogen with a mass of 1 is replaced with deuterium with a mass of 2, it should slow down quantum tunneling, because tunneling is very sensitive to mass. So what you can do is do these enzyme reactions in both water and in heavy water, in deuterated water. And you measure then the kinetic isotope effect, the activity of the enzyme in ordinary water compared to the activity of the enzyme in deuterated water. And if that activity is different, you have a kinetic isotope effect. Doesn't necessarily mean it's quantum mechanical, but by doing a lot of other experiments, you can demonstrate it is quantum mechanical. And for example, Nigel Scruton at the University of Manchester published this paper. It hasn't got the date here, but I think it was about 6 or 7 years ago in Nature. So what they are showing is that this is a classical double well potential, and in this reaction, this is the particle which has to, if it's going classically, needs enough energy to jump over this barrier to get into the other well the product well of the double well potential. And normally it won't have that energy, but quantum mechanically it may have that energy. It may be able to do that by quantum mechanics by tunneling through this barrier, but that rate of tunneling differs between hydrogen, deuterium, and tritium. So by measuring the kinetic isotope effect in water, in deuterated water, and in tritiated water, and seeing how that is compared to how it would be a classical over-the-barrier mechanism or a quantum tunneling mechanism, they demonstrated that this enzyme achieves its catalytic efficiency through quantum tunneling. And that's been demonstrated for many different enzymes now, that as well as aromatic amine dehydrogenase. Most recent I saw was only a few days ago, when the enzyme involved in making peptide bonds in ribosomes. So the enzyme that strings together every single protein that has ever been made involves quantum tunneling of protons. So a key feature in how enzymes work is they bring substrates together in such a way that it allows particles to quantum tunnel from one place to another. Essentially, it's kind of the particle disappears at one point in space and appears at another point in space without visiting the in-between places. This is what tunneling is about, really, quantum mechanical tunneling. It's a peculiar quantum mechanical phenomenon, and it takes place inside enzymes. Not only electrons tunneling in enzymes, but proton tunneling in enzymes as well. So that's one of the other aspects of quantum mechanics that seems to be involved fundamentally in many biochemical reactions. Now we'll look at, I think I just look at this one more, which is avian navigation, which is one of the most fascinating stories in quantum mechanics. Our familiar robin, bobbing around in our gardens at the moment, is not the substrate of this talk, because actually it's the European robin. The British robin does not migrate, which is why it stays around to be with us in the snowy Christmas time and ends up on our Christmas cards, because it doesn't migrate. The European robin in Scandinavia and Northern Europe migrates south in the winter. Looks just like our British robin. This group of ornithologists, the Johnjoes, Wolfgang and Roswitha, demonstrated that the birds use a compass. Now, bird navigation, in common with other animal navigation, usually is multifaceted. There are many different cues that animals use to navigate. They use clues of the landscape. They use, some of them seem to be able to see star fields and navigate by them. But many animals have a compass. And that's been known for a long time. And that's peculiar in itself. How do you make a compass in a person, or in a robin, sorry. We don't seem to have a magnetic sense. But robins and many other animals do. How do you make a compass in flesh? It's a tricky thing to think of how you would do it. And what the Johnjoes showed is that the compass that robins have is peculiar. Well, it's not peculiar to animals. Many animals have this, but it's not like a Scout's compass. It's not like a simple magnetic compass that we'd use for trekking around the Lake District. It requires light to work. And it's an inclination compass. It finds the nearest poles, but cannot distinguish between poles. So unlike a Scout's compass. What is an inclination compass? What an inclination compass does is it detects this inclination angle of the Earth's magnetic field against the surface of the Earth. So this shows the lines of the magnetic field of the Earth's magnetic field, and at the poles they're vertical, at the equator they're horizontal. If you can measure this angle and point towards it, then you can end up going to either the North Pole if you're in the Northern Hemisphere, or the South Pole if you're in the Southern Hemisphere, but you can't distinguish between them. So an inclination compass points at the nearest pole. Wherever that is, you can find your way to or away from it. But it doesn't distinguish between a north or a south pole. So those were the two properties that the Wilczkows found about the Robbins compass. It required light and it was an inclination compass. How does that work? Now, there's a long story to this, and I'm just showing you the punchline. It's given the time, it's now 8 o'clock. This guy came up with a model. In fact, it was a chemist, Schultz, who first came up with this model, and Thurston Ritz, who was a PhD student in his lab, came up with a more sophisticated version of it and attached it to a protein called cryptochrome. And this paper was published in 2000, a model for photoreceptor-based magnetoreception in birds. Cryptochrome is a photoreceptor in our eyes and also in robin's eyes. It's a protein that will pick up light and detect light. And he proposed that the compass is due to entangled electrons that can detect the Earth's magnetic field. Now, what do we mean by that? So this, what he proposed was that the compass is based on the generation in cryptochrome of a radical pair. Now, we didn't know what that radical pair was, but a radical pair is essentially generated by various chemical reactions in which you have radicals which have unpaired electrons, and if you have a pair of those, what's called a singular or a triplet pair, then they can interact, and when they interact, they can detect a magnetic field. So they oscillate in a magnetic field, and if they interact and form a compound, then the rate at which they form this compound will be affected by a magnetic field. So he proposed that in the cryptochrome molecule were generated radical pairs that interact because they are quantum entangled. Now, there isn't time to go into the whole theory of this, but essentially this only works if you generate free radicals that are quantum entangled. Now, his supervisor, Schultz, showed that this works in chemical reactions, that you can generate some chemical reactions are sensitive to magnetic fields, and they are because they generate entangled radical pairs. What Thurston Ritz claimed that this was happening in the bird's eye in this cryptochrome molecule, and that these radical pairs were generated that are interacting with the Earth's magnetic field. And the model predicted that if this was going on, so the model is that in the bird's eye is this cryptochrome molecule. When it picks up a photon of light, it forms a pair of free radicals that are quantum entangled. That interacts with the Earth's magnetic field to generate products. And the products, whether they're from singlets or triplets, will depend on the magnetic field. So the bird will be able to see the magnetic field in its eye through the generation of these free radical pairs that are quantum entangled. So a very speculative proposal, but it made a prediction that the bird's compass should be disrupted by weak external magnetic fields. And he paired up with the Wilczkows to test this with birds. So they took robins and tested whether they could disrupt their compass with radio frequency magnetic fields, and indeed they could. So the model predicted that the bird's compass should be disrupted by high frequency radio waves, and indeed it was. And that's because the high frequency radio waves are disrupting this interaction between the entangled radical pairs. So this was then published in 2004, evidence that the bird's compass is quantum mechanical. Now, there's still an awful lot of research to be done in this area, but at the moment, there's no model that can account for this without it being quantum mechanical. So although it doesn't prove that it's quantum mechanical, there's nothing that really fits the experiments except the quantum mechanical compass. So now there are many other areas of quantum mechanics, of quantum biology that we could go into if we had time. Quantum mechanics, as the earlier slide I illustrated with, has also been implicated in our sense of smell, that our sense of smell may also involve quantum tunneling. Luca Turin has proposed that and found strong evidence for that. Consciousness has also been proposed, and there's not really any evidence for that, of course, but there are many other areas which seems to be quantum mechanical phenomena are coming up in living systems. Now, what I wanted to also tell you about is our own interest at the University of Surrey, mine and Jim's, which led to the book that we published. So actually, you all know where Guildford is. This slide is to tell people where it is. But the interest came because I'm, as John was saying in the introduction, I'm really a microbial geneticist at the moment. I started off in biochemistry, but now I'm a microbial geneticist. And in 1988, this paper was published by John Cairns at Harvard, a very eminent microbial geneticist, worked with Watson and Crick at MIT and at Harvard, and he published this paper, The Origin of Mutants, which suggested that mutations could become more frequent when they provided an advantage. And this was a big thing. It generated a storm of controversy in genetics, not only microbial genetics, but all of genetics, because it's almost Lamarckian in that it allows mutations to become more frequent if they provide an advantage. It's as if the organism could choose how to evolve, where to evolve. The experiment he did was quite simple. He took E. coli cells and grew them. Well, he didn't. He didn't grow them. He took E. coli cells that he'd grown and put them on two plates. One which had a substrate that they could grow if they could mutate, and one which didn't have that substrate. And what he found was that the mutation rate to allow them to grow took place much more rapidly on the plates in which the substrate, which was glucose, was there. So his experiments suggested that cells may have mechanisms for choosing which mutations will occur. Extraordinarily controversial at the time. It generated thousands of letters to Nature. But the experiments were sound, but there's still lots of controversy about these experiments, and most people believe there are other explanations than what I'm going to talk about. But at the time, I had read John Gribbin's book, Schrödinger's Cat, and that got me onto Schrödinger's book, who claimed that mutations represent some kind of quantum jump. So Schrödinger, if you remember, back in 1944, published What is Life, in which he suggested that life was dependent on quantum mechanics, and he suggested mutations were some kind of quantum jump. Now, if we look at a base pair, what you should notice, of course, is that the base pair is held together by hydrogen bonds, and hydrogen bonds are essentially shared protons. So they're fundamentally quantum mechanical entities. That proton is shared between an oxygen and a nitrogen atom. That proton is shared between two nitrogen atoms. So they're fundamentally quantum mechanical in nature. This is our genetic code, really. Where these protons are determines our genetic code. It's quantum level coding. This guy, Pierre Olaf Lodin, proposed back in the 1970s that DNA code is essentially a quantum code written on proton positions. Essentially, this is the DNA code. DNA code is these proton positions. And he proposed that mutations may be caused by protons tunneling into the wrong position, tautomerization. So this is an AT base pair. This is the protons that are involved in the hydrogen bonding between the base pairs. What he proposed was you had a double proton transfer in which this proton popped over to here and this proton popped over to there, like that. So you had a double proton transfer. Why you needed 2 protons was to maintain the charge across the bases. So he proposed this reaction may cause the formation of a tautomeric form of the base. And in the tautomeric form, the normal form of the DNA bases is the amino forms, but if they're in their tautomeric form, then instead of having the canonical Watson and Crick base pairs, thymine pairing with adenine, cytosine pairing with guanine, you can have mutagenic base pairs in which cytosine will pair with the imino form of adenine. So if this was happening in incorrect base pairing, could then cause mutations. And this is what Lodin proposed. What I thought, well, if that was happening inside John Cairns' experiments, then it could be involved in how the mutations became more frequent when they provided an advantage. Now, I then gave a talk at the physics department I phoned up someone in the physics department and said, oh, I've got this great idea. And they said, well, come along and talk to us about it. So I gave a seminar, biologists giving a seminar in the physics department about quantum mechanics was a pretty dangerous thing to do, and I was justly received with a lot of skepticism, quite rightly. But Jim Al-Khalidi was in the audience, and he came up afterwards and said, well, it doesn't quite work really what you're saying, but maybe we can work on it. And we did over the next year or two, and we'd meet over a coffee or a beer, and we eventually came up with this quantum mechanical model of adaptive mutation. Now, I won't go into it. It's a kind of, to be honest, we've kind of abandoned it as a model of adaptive mutation in which this decoherence, the quantum measurement, will actually affect the mutation rate. I then published a book back in 2000 called Quantum Evolution, in which I expanded on this. But we've kind of, adaptive mutations became kind of messy in microbial genetics. So we've left that and just gone to the question of whether quantum tunneling is involved in mutation. And this is some work that Jim's done now looking at theoretical density function calculations to look at whether quantum tunneling is involved in this transfer in tautomerization, in going from the normal AT base pair to a tautomeric form, which is mutagenic. It could go through classical processes, jumping over the barrier, or it could go through quantum tunneling. And he used density function theory to look at that, and I won't go into the details of it because I can't understand the details of it. The protein can be excited over the barrier by molecular vibrations, by heat, for example. This shows the proton is going from one place to another. This can be considered the normal base. This can be considered the tautomeric base. It can do it by hopping over the barrier classically, or it could go through by a tunneling mechanism. And the question he addressed is which goes on. And he did it by the density function theory, and he came to these conclusions. The environment provides thermal assistance to protons to protons to tunnel. So if protons do transfer to the tautomeric, essentially what he showed was the only way that the protons were getting from one place to another was actually through the tunneling reaction. So at least in his theoretical calculations, this tunneling reaction was taking place, but it required heat. It required thermal activation. First of all, the electron had to be raised to a higher energy level, and then it could tunnel through the barrier. So this going over the barrier didn't seem to work. It did seem to only go by a tunneling reaction, but one that required thermal assistance. Okay, so that he published that a couple of years ago. Okay, so that's the theoretical side of what we're looking at the moment. The other side of it is seeing if it happens in the real world. So in my lab, we grew E. coli in ordinary water and deuterated water. Remember back to the enzyme experiments, deuterium should slow tunneling, and we measured mutation rates. This is E. coli, the dots are mutants, you count them, and this is what we got. So in water, 50% D2O and 100% D2O, there seemed to be a depression of mutation rates. Big error bars, though. So I'm not really sure if I believe it entirely. We've since done it by doing the PCR reaction. There's also too many things going on with whole E. coli cells. So we did it with just the DNA polymerase reaction, did the same kind of thing, but now just in a test tube, replicating DNA with the enzyme DNA polymerase. And we got a similar depression of around about 40%. So it's consistent with tunneling, but it still doesn't really prove tunneling. We have to do a lot more, but it's interesting. So does tunneling cause mutation? Maybe. So that's it for my review of quantum biology. I'll just mention one other thing, which may be of interest. We've now established, with funding from the Leverhulme Trust, after doing nothing but chatting over a cup of coffee or a beer over the years, we've finally got some money to do some quantum biology at the University of Surrey. The Leverhulme Trust gave us the funding to establish a doctoral training center in quantum bioengineering, involving a lot of different scientists at the University of Surrey, looking at photosynthesis, tunneling, and all sorts of quantum theory and all sorts of other areas of biology that were looking for quantum mechanical effects. This is what we've got: 5 years of funding, 21 PhD students. We've already started with about 8 of them this year. We've got a number of different projects which look at proton tunneling, photosynthesis, biological noise and quantum coherence, avian navigation. I won't go into the details of them, but there are many different interesting projects that are going on at the moment. And this is our team at the moment. There's Jim and myself. And why I wanted to put that slide up is that we are looking to collaborate. We are looking to encourage PhD students to come, to spend time in our labs, to generate, or to maybe with other institutes like Imperial College, to have collaborative PhD projects in which we have, say, 2+1, 1+2, in which we can work on biology across different institutes across the UK. So we'd just like to collaborate with anyone who's interested in collaborating. So does quantum mechanics, and the other last thing, so drugs and medicine interact with the body at the molecular level. We know that already, and we now know that this interaction involves quantum mechanics. So biology needs quantum mechanics, is the last word I'm going to say on the talk. Thank you.
Thanks very much to Johnjoe. I might take a moment to draw a breath there, because there's an immense amount in there. I'm sure there will be questions, and there are already. So we'll have our runners with our mics. Bill's got both of them. So here we go with the first one.
I'm interested in the possibility of quantum mechanics and the light and heat aspect, and with skin mutation, in particular leading to skin cancer.
Yeah.
For instance, I have a friend whose doctor said she mustn't ever be in the light or again, or under the sun. And one other question, because I did study music, is do these chemical reactions make sounds? Has anybody ever thought of measuring that?
Make sounds. Well, chemical reactions, no. I don't think necessarily. I mean, the sound is a vibration in the air, and they don't necessarily make a vibration in the air. You could, of course, generate some, a chemical reaction. If you have an explosion, bang, that'll make a sound, and that's a chemical reaction. So they can make sounds, but I wouldn't say it's kind of fundamental. The first question is much more interesting, I think, is whether they're involved in cancer. And this is why we're so interested in the question of whether quantum mechanics may be involved in mutation. Mutation is, of course, responsible for evolution, but it's also responsible for cancer. So if tautomerization of DNA bases by proton tunneling is involved, then we need to know about it in order to know better how to avoid mutations that can lead to cancer. So it's a very important question. We don't know the answer to it yet.
Okay, over to Alan here, and then we've got 2 there, and one behind you, Bill.
Thank you. Very interesting talk.
Thank you.
Can I offer a comment and also ask
Sure.
a question? The comment was that I seem to remember reading that studies of families in terms of their DNA, measured repeatedly to give a very accurate readout of their DNA, and their children, so mother, father, and child, showed that typically there was an error rate of about 30 SNPs, single nucleotide polymorphisms.
Yeah.
It would just be interesting, so my comment is, it would just be interesting to know if anyone might attempt to relate the models that you're advancing for mutations in DNA replication,
Yeah.
or similar replication.
Yeah,
of
it
RNA,
is.
could lead to that sort of. I mean, it's an extraordinary. Strikes me at least as an extraordinarily good reproduction rate.
Yeah, no,
fidelity
it's
of 1 in 10 to the 8.
the fidelity of DNA replication is extraordinary. As you say, 1 in 10 to the 8. It should be a lot higher than that. If tautomerization, if that was all that was going on, tautomerization, say, then it should be a lot higher. It should be, say 10 to the minus 3 or 10 to the minus 4. But we know that within DNA polymerase and all of the enzymes involved in chromosomal replication, there is a huge amount of error correction machinery there. Yeah. And that reduces the mutation rate to those 10 to the minus 8 kind of levels. So the question is obviously it can't reduce it to 0, otherwise we wouldn't get any mutations and we wouldn't be here because we wouldn't have had evolution. But it's the question, yeah, it could be. It seems to be that the estimates of what the mutation ought to be with tautomerization is so much higher that the low frequency of mutation that we find in living organisms is mostly dominated by error correction machinery. So there's not much point in trying to look at the tautomerization rate to account for it because it's so affected by the error correction machinery.
Yeah, fine, thank you. And my question, if I may, is in your description of photosynthesis,
Yeah.
you described how the absorption of a photon from light, typically in the visible or near UV, i.e. 2 to 3 or 3.5 electron volts
Yeah.
per photon, leads to an exciton,
Yeah.
which can migrate through the chlorophyll and
Yeah.
so forth. But I seem to remember reading that actually that isn't enough energy to do the job. And it's actually a 2-photon process, is it not?
Don't think so. I don't know of that. Any photosynthesis
Well,
expert in the
drive
audience?
the reaction where the plant
Oh, the
absorbs.
reaction center, in order to fix that energy, it needs more than 1 photon, yes. When it gets to the reaction center, it needs several photons to make a molecule of ATP. I can't remember how many. And that's how the energy is fixed when you make a molecule of ATP and some electrons for reductive processes in the reaction center. So the transport involves single excitons, but in order to capture that energy into a chemical, yes, you need several photons to get there to the reaction center.
Fine.
Yeah, you're
Super,
right.
thank you.
Yeah.
Okay, over here now, Johnjoe.
Hello. Can you say something about quantum mechanics and consciousness, which is on your
I
list, and
could
what
do,
its
yes. So this
implications?
is obvious, always comes up in questions. So the idea of quantum mechanics being involved in consciousness was, I think, well, actually goes back way before Roger Penrose. Actually goes back to the Gestalt psychologists back in the 1920s. But Roger Penrose famously teamed up with Stuart Hamerhoff, the anaesthetist, to make the claim that consciousness is fundamentally quantum mechanical. And they made a model in which the microtubules inside cells form quantum coherent structures across the entire brain. No physicist I know thinks that that is the least bit feasible. The kind of thing that which I've been talking about today, say the coherence inside a single photosystem, is already stretching credibility for physicists. They say, oh, no, it couldn't really happen, but it does, which is great. But then to say that coherence can extend across an entire brain, so rather than the nanometer scale, the centimeter scale, no physicist I know would believe that that could be possible. So it still gets an awful lot of interest, quantum mechanics, and I've never understood what is the what is the justification for putting quantum mechanics in the brain? It seems to be that kind of we don't understand quantum mechanics, we don't understand the measurement problem in quantum mechanics, we don't understand consciousness, so they must be related. And I don't think that's as good as it gets for the argument for quantum mechanics being involved in consciousness. It may be, but I don't think there's any good argument, and certainly the model that Penrose and Hammerhoff have put together doesn't make any physical sense.
Okay. So I think I'm probably going to be a spoil sport tonight. We've got one there and then one at the back. Time's the enemy, isn't it, unfortunately? So if I could beg forgiveness after that, then we'll be able to get the evening with the usual wine, etc. And for those who are over 18, obviously. Okay. So first of all here.
Thanks. So let's assume quantum tunneling causes mutations. And quantum tunneling follows a probability distribution, so it's kind of random.
Yeah.
Then how could an organism increases the probability of useful mutations?
You have to read our 1999 paper. Essentially what we're claiming is something like what's called the inverse Zeno effect. So as you say, quantum tunneling is a probability curve around it. So if you imagine that probability curve, a kind of Poisson-shaped probability curve, if you measure at the extreme end of that curve, the only thing you can possibly measure is a particle that's got to that place, okay? And you can capture it then. If you measure it and make it classical in some way, you will capture it there. And if you are doing all your measurements there at the extreme end of that distribution, then you can drag all the particles that are in your system to the extreme end, because that's the only place where they become classical. Do you kind of see that?
So it's basically about the locality of measurement?
Yeah, you're essentially measuring only when the particle is in the state you're interested in. So it's like if you have the double well potential, so you have the particle zipping between both, say, going very fast between the two wells, and you're looking only at the one of the wells, and you say, when I detect a particle in this well, I pull it out. And imagine now 1 trillion particles who are bobbing backwards and forth in this well, and you're only looking in well B. And you say, every time I measure and find, sometimes I look in well B, it's not there. Sometimes I look, it's not there. But when it is there, I pull it out. Eventually, for your 1 trillion particles, because you're only pulling them out of well B, they'll all be in that state. It's a kind of difficult one to get over in a few sentences. If you read the 1999 paper, you'll get a better feel for it, hopefully. Or we can maybe talk a little bit more about it over a glass of wine. I'll try and explain it then.
Thanks a lot.
And the last one, unfortunately, I'm totally blown away by the quality of the questions. You know,
they are tough questions.
I couldn't have thought of any of them.
Is the mic working? Yeah. Photosynthesis and the travelling salesman issue.
Yeah.
That immediately made me think that maybe quantum computation can solve the NP-complete problems.
Yeah. Yeah, that's been speculated a lot. No one, until we can make a quantum computer, and I'm sure scientists here in Imperial College are trying to make. There are some quantum computers around, but they're not very clever so far. But I think that is one of the ambitions to be able to solve insoluble problems like the NP problem with quantum computers. You should be able to do it because you can explore far more states. And this is where, Seth Lloyd, the guy who came up with the Goldilocks hypothesis about photosynthesis, he's a quantum information theorist. So he's interested, his lab wants to make quantum computers. So, and the great thing about, if everything is right about what people are saying about what's going on in photosynthesis, that it's quantum coherence, it's performing some kind of a quantum walk, a quantum computation of some kind, then it shows that you can do quantum computation at room temperature in large macroscopic systems. If you can do that, then you can make quantum computers that will work on your desktop. And that would be such a remarkable thing, because the power of quantum computers exceeds, one decent-sized quantum computer would exceed that of all the computing power we have in the world at the moment. So if you can make them using the lessons that life has somehow maintained, and I didn't go into this because of the time, but the peculiar thing about life seems to be that whereas if you perform a quantum mechanical experiment in your physics lab, you remove all vibrations. You perform the experiment in a vacuum, you cool the system down to absolute zero or as close as you can get to it, you shield it from vibrations, and then you can detect quantum coherence, quantum tunneling, entanglement. So you remove vibrations. The very peculiar feature that's been discovered about the quantum mechanical features of biology is that they need vibrations. So instead of shielding the system from vibrations, actually most of the quantum mechanical phenomena in life are vibration assisted. So somehow life has turned the decoherence thing around. So instead of decoherence, random vibrations destroying coherence, they actually maintain coherence. And this may be the most peculiar feature of how living cells work that may be very fundamental to life, and it could be a remarkable thing to take forward perhaps into quantum computing.
So I knew we were going to end on a high point like that.
Okay.
Thanks very much to everyone for your attendance.
Thank you.
Johnjoe, I can't thank you enough. It's a brilliant lecture.
Pleasure to be here.
And let's show our appreciation in the usual way.